So you will have an announcement just now. We recorded simply to keep a record and also for security reasons, and also just in case some participants or people who couldn't make it want to watch it afterwards. So hello everyone, and welcome to the Sanger Institute. I will be taking you through. I will start while people are trickling in, just to make sure that we don't take too long. So my name is Alena, and I am here with colleagues Damien and Louise. If you can wave so people can see you. We are part of the public engagement team at the Sanger Institute, and we will be hosting you through your visit to the Sanger cellular facility and human cell atlas laboratories today. But and I'll introduce the scientists that will be doing that to you as we go along. Now that's us. We would like to know about you. So if you could pop in the chat, where are you joining us from? That would be fantastic. We would love to know where you are all connecting to us from today. Oh, it's great. So answers are coming in. It's pretty much all over the UK. We have someone in Geneva, London, Brighton. Oh, this is great. So pretty much throughout the UK, which is fantastic, and some from abroad as well. Belfast, Cambridge, Leeds. That is very nice to know. Thank you very much for that. I will also run a little poll to find out more about you. So if you could answer on your screen whether you're a teacher, whether you're a student, school, university, whether you're an artist, or whether you're a member of the public who loves science. There we go. So we can see that. Can you see it? So we have a mix of people. School students won, most of them. That's great. Thank you very much all for coming. And now, without further ado, I will share my screen. And I need to close these things down. Can you see my screen?
We just have a rainbow.
Yes. Rainbow, yes,
yeah. That's fine. Yeah, that's great. That's what it's supposed to be. So if these were normal times, you would be coming to the Sanger Institute in person. And to do that, you would turn off the main road and come to the entrance here, and then turn into security right behind the tree to get your badges that would allow you on site. However, these are not normal times, so never mind security, just come with me, I'll take you through. You would arrive at what we call the plaza, where you have the restaurant on the right here, very important. And then on the left, you have the Morgan building, which is one of the buildings of the Sanger Institute. And as you walk down the path here, right in front, you see the Solston building, which is one of the main buildings of the Sanger Institute, where most of the laboratories are. Here on the right, you catch a glimpse of the EBI, or European Bioinformatics Institute, that is also based on the Genome campus. As you go down through the path, you see the lovely gardens that we have on site, and you reach Hingston Hall. Kingston Hall is the stately home that all these grounds belong to, and was designated the site for a new research institute to participate in the international consortium called the Human Genome Project, whose aim it was to sequence the human genome. It was funded by the Wellcome Trust, and this is it, as it was then. It was called the Sanger Center after Fred Sanger, who came up with the technology to sequence DNA. And it was under the direction of John Sulston, who set immediately to the task of sequencing the human genome, but also at the same time sequenced smaller organisms just to set up the technology and the expertise. And so it was that in 1996, only 3 years after opening its doors officially, the yeast genome, all 12 million bases of it, was sequenced, followed in 1998 by the genome of tuberculosis and the whole genome of the worm C. elegans with all 100 million base pairs of it. And also at the same time, 90 million of the human genome bases. These are the insights of the old Sanger Center. So having thrown all of those numbers at you, can you put in the chat, how big do you think the human genome is? And someone will have to tell me what's coming up in the chat because I can see it. Are there any answers coming in? No? Come on, I guess. No
I've got
guesses?
a couple of answers for 3 billion.
Yes, that is correct. It's 3 billion bases. So of course, not a trivial task to sequence. Nevertheless, the first draft of the whole sequence was published in 2001, and the full genome was published in 2003, with the Sanger Institute being the major single contributor to that sequence. Of course, the work is not done because there are still bits that have not been sequenced completely, and then we don't understand all of it and how it works. So this is the old site as it was then. It has evolved enormously since those days to the enormous site that we have today with all the buildings here. You can see the Sulston building as it is today. And the work goes on. So I'll take you now back to the Sulston building. You can see it here on the left. And with a glimpse of that, I will leave you at the reception of the building, and I will pass the baton on to Maria and Adam, who are two researchers from the cellular genotyping and phenotyping facility at the Sanger Institute, to explain to you their work.
Okay, so I'm just going to load up this video now. So can everyone see this? Is that sharing well? Okay. So good afternoon, everyone. So today we'll be starting you on a virtual tour of the CGAP labs. So we're a core facility that provides central cell biology support to the Sanger Institute. So we function mostly as a contract research group in partnership with faculty groups to carry out multiple distinct and often large-scale cell biology projects. Some examples of our work include looking at genes that are essential to cancer cell survival, and then causes of rare diseases and developmental disorders as well. So today we'll show you around the lab, and then you can see what we do on a day-to-day basis. So we're a category 2 level lab, so this means that we have to wear PPE to protect ourselves when we arrive in the lab, and we also have to always put on our lab coats and protective glasses and wash our hands as well. So in the current situation, to be COVID secure, our lab coats are currently kept in plastic bags to minimise the risk of them touching other lab coats. And we have strict rules in place around the lab, such as room capacities, cleaning down workspaces, and touch points after use. So I'll now hand over to Maria, where we're going into the quarantine labs now.
Thank you, Adam. So the first lab we're going in is our quarantine lab, where we receive the tissue samples from real patients. These samples could be a tumor biopsy, and if you could stop the video there a little bit, Adam, please. So yeah, these samples could be a tumor biopsy, skin or blood samples. And before they pass into our main labs, they must first get tested negative for mycoplasma, which is a pathogen. It's harmful for human, and also it can ruin our cultures. So most of our work is done in a safety cabinet, like the one Laura is working in here. Here, Laura is deriving the colorectal organoid cell line taken from a biopsy from a cancer patient. So you can continue now the video, Adam, please. So this particular tissue sample is being broken down into single cells, and then they will be cultured in a 6-well plate, which I'll show you in a minute, with the hope that we can generate a new cell line that can be later used in different research projects by the faculty teams. We do this by trying to keep the sample in conditions as similar as in the human body as we can possibly do. So here those cells are put into a gel-like substance called BME. Is this pinky fluid that you can see now. It's called basement membrane extract, and it gives them like the scaffold they need, the cells need to grow into 3D organoids. And this plate will be then incubated at 37 degrees, which is the same temperature as the human body. This is the incubator where we keep our culture. Then we check the cells and we see if they grow.
Okay, so we're now heading into the core lab. So this is basically the central space where we can all meet and do sort of general things. So within the core lab itself, we also have our histology section, which I think is going to be talked about a bit later as well. But histology, just briefly, is the study of human cells and tissues beneath a microscope to discover their structure and function. So we work on a project called the Human Cell Atlas, or HCA, which is trying to create a full map of the human body. We're very fortunate to receive donated tissues from patients to work on. Part of our work involves cutting this tissue down into very small sections, as you can see Monica doing here, and this is done on a cryostat. So this tissue will be frozen down in a block and fixed to a chunk on the cryostat, and this is held at temperatures between -15 and -25 degrees, and this helps us to preserve the tissue. So once a section has been cut, the tissue is put on a slide to be stained, and then this staining allows us to see these cells beneath a microscope and investigate them further. So yeah, you can just see Monica in there now, slicing. And yeah, like I said, they'll be talking about it in a bit more detail a bit later on as well. So yep, so she's just placing a very small section of tissue onto the slide there and then warming it so that it can attach properly. One of the other machines we have in here is called the Leica Bond. So the bond stains the slides with fluorescent dyes to detect genetic material within our cells. The bond requires some manual preparation, but it automatically runs overnight. So here you can see Sophie register the reagents on the machine and make up the mixtures of fluorescent dyes before she puts these into the machine. So here are the alcohols, waste bottles, water, and important solutions that would be needed for the bond to run. So yep, so she's just processing them now, and then yep, loading them in. And then yep, there you can see all the waste materials I just mentioned earlier. And then this robotic arm here will scan all the reagents ready for the overnight run. So it can just be left and then it will carry on overnight. And hopefully they'll get the results. So yeah, there's also a lot of equipment in the labs as well, as you can see. So a lot of it is even mind-blowing to a lot of us. So we do lose track sometimes. So here we go back into core. And then from here, we're going to be moving on into the media lab now. So I can hand over back to Maria.
Yeah. So the media team is an essential part of our department. The media, for those that doesn't know, is basically the food we give ourselves to keep them alive and happy. Cells are very delicate and difficult to grow in the lab, as you can imagine, and they need the exact right amount of nutrients, sugars, growth factors, and concentration of gases like oxygen and nitrogen. So if you could stop the video here a little bit. So therefore, because they're so delicate and so specific, the media is a key element, not only to keep the cells alive, but also so they can grow, differentiate, or produce organoids, which we saw earlier on. Different cell lines need different types of media. So each media has a number of components that is specific to that cell type. Here we can see Wes preparing some wind condition media, which will be used to grow colorectal tumor organoids. And if you can carry on with the video, Adam, now. So here Wes is filter sterilizing the media, because the media has to be sterile to avoid any sort of contamination to the cells. Contaminations can kill the cells very easily. Another service the media team provide is the mycoplasma testing for the samples that they arrive into our quarantine lab. Plus the sheer scale that the media team works is pretty incredible, because they produce bucket loads of this media for every single project. I think we calculated a while ago that only for a single project they were producing so far like 7 full bathtubs of media. That's a lot of media. Yeah, so back to you, Adam.
Okay, so the next lab we're going to head into is where we work with some cancer cells. So we're just going in there now. So this is one of the other larger labs that's linked to that core lab. So we can culture cells from lots of different types of cancer in our lab. And these are great models for helping us to gain a better understanding of the disease and how we might potentially treat it in the future. So one of our responsibilities as cell biologists is to make sure our cells have the right conditions to grow nicely. In this clip, you can see Emily adding fresh culture media to a plastic bottle and then mixing it up a suspension of cancer cells and then adding this to the media. So she's given these a really good mix together, and then together she's going to pour them into this cell culture flask. The reason she's doing this is because cancer cells continuously divide and grow, and then they can also get too crowded as well in the flasks that we grow them in. So we have to take a portion of these cells and then place them into fresh culture flasks to give them more space to grow. So that's just one flask, but we will sometimes, have hundreds of these things just to give the cells physical room to grow. So this process is known as a passage, and cells may require a passage 1 to 3 times a week, depending on how fast these cells grow. So yeah, Emily's just moving it back and forwards just to get the cells there. So we've just included a picture here to show you what some of these cancer cells will look like under the microscope. So this could be the same line. However, on the right, this would be a line where, as I mentioned, they're really bunched closely together. So we need to just break them apart so that they can have the physical room to grow across the plastic. So even though you can't see them with the eye, they can really take over all of that area to keep on growing. And then, yeah, on the left there would be an example of a line that we will have just put into a flask. So you can see they've got all the room to grow, but sometimes they can just keep on spreading out. So I'm just going to move you on to the next project that we have, which is similar, which is the CRISPR-Adams project, which Maria can talk about now.
Mute, sorry. Now, cancer cells require fresh media 2 to 3 times per week to give them the nutrients they need to grow. So here you can see Verity changing the media in a multi-layer flask using a media pump. The large amount of cells we grow, so we have to find a solution because of the larger scale of cells that we grow. And we grow them in these multi-layer flasks. So this is a 5-layer flask. So that means that the cells have like 5 times the room to grow. So we need 5 times less amount of flask, even though the amount of flask that we need to grow all the cells is amazing. And you're gonna see here on the incubator. It's a lot of flask that they need media changed every other day, basically. So that's why we use the media pump, because it will be like extremely ergonomically discomfort, like to do it by hand. So yeah, this is another large project that we use.
Okay, so just heading around the corner from there, and to follow on from the cancer lines that we were talking about, and the organoid derivation lines that we've already seen, is one of the projects that we use organoid lines for. So just briefly, organoid lines are a type of cancer line that we grow as well. However, they've all been developed within the department straight from tumour samples that we've received. And they, in theory, will mimic exactly how these tumours will behave in the body. So we can grow them in 3D, and they're essentially mini tumours that we can grow, and they'll be, hopefully more identical to tumours that would be in the body. So you can see on the screen here, you can see me looking at these tumour models. So this is just in theory replicating what we would see in the colon or esophagus. It's quite hard to see on the screen, but you can see under the microscope, these are all essentially mini versions of what we would see in the body. So these models are really useful for screening potential new drug treatments, and within the department, they're used for studying any important genes that these cancer lines might need to survive. And this can help us understand how well the best way to treat these cancers could be, because we're essentially doing these experiments on what would be in the human body. So in the video here, you can just see me prepping these samples as one of our quality control checkpoints of our pipeline. And this is just to make sure that each stage of our pipeline is going correctly and how we would expect. So yeah, it can be quite repetitive at times. So you can just see me just sort of prepping a lot of these samples ready to go for further analysis. I think that is definitely sped up. I'm not that fast. But yeah, so there's lots of stages within all of our projects, and they can all be quite diverse as well. But yeah, it's quite good in that respect. So the next one is parts Hipsky, which is back in the quarantine lab with Maria.
Yeah, if you can leave it paused there a little bit while I introduce. So another big part of the work we do in CGAP involves working with induced pluripotent stem cells, which can be also known as iPS cells. Here you can observe what some of not confluent iPS cells look like as they were passaged the previous day. So this is what we were talking about. Once we split them and we'll give them more room to grow, they look quite sparse. This is how no confluent cells look like. So yeah, you can carry on now. Stem cells are a very unique type of cells that can become specialized into any cell type in the body. CGAP was initially set up to run a project called Hipsky that aimed to create iPS cell lines from normal and diseased patient samples. And is at the moment is one of the world's largest characterized iPS cells resource we have. In this video, you can observe Rebecca media changing the iPS cells you saw previously. These cells are very, very extremely delicate, so they need media change every day to ensure that they grow healthy and they are happy. We also are using stem cells like this for gene editing experiment. Using CRISPR technology, we can knock out every gene in the genome to find out if it's essential, and then discover whether these essential genes are the same in everyone. So now we're going to show a few pictures of the cells growing on the flask. Well, in this case, on a plate. So these are the induced pluripotent stem cells. So you can see that they grow on circular colonies with smooth edges. And then the next picture will show dopaminergic neurons. So these are dopaminergic neurons which have been derived from stem cells. And the next one show cortical neurons also derived from stem cells. And I forgot to mention that one of our projects aims to derive neurons from these stem cells of real patients to compare how mutated neurons found in disease compared to healthy neurons. And that's over to you, Adam.
Okay. So as you've seen throughout this video, most of the work we do is carried out in these microbiological safety cabinets, which we all just call hoods. So these are there to keep our work protected from the outside environment, but also they'll protect us from any substances that we might be working with. And then in order to use these, we have to maintain sterile conditions. So we need to work in a way that reduces any risk of contamination. So we clean the hoods before and after use. And as you can see here, James is cleaning down the surface of the hood when he's finished. And then when we finish as well, we'll shut them down completely, and then we'll turn on UV lights to further sterilize the inside of it. So yeah, we can be a bit over the top with keeping them clean, but yeah, it's very important to make sure our work remains intact.
And here is me in the LN2 room. So all the cell lines generated in CGAP are frozen down and stored in little cryovials until they are needed. Because these cells are very delicate, we need to store them, we need to freeze them in liquid nitrogen. So these tiny cryovials are put in these boxes that you can see here. Then the boxes are stuck into these racks, and the racks, they go inside the tank, which contain liquid nitrogen vapor, which keeps the temperature at minus 192 Celsius. So it's pretty cold.
Okay, and then yes, that's it for our lab tour. So this is just an example of some of the projects that we have running in the department. You can all look them up on the website as well. On the Sanger Institute website, they should have all had information. And yeah, a lot of these projects are part of a lot of work and a big team as well. So there's a picture of all of us on there working from home. But that's just sort of one page of the team. So there's a lot of us working in these teams. Because yeah, a lot of these even smaller projects will take so much work. And then yeah, so me and Maria will be more than happy to take any questions that you might have. And then yeah, there's just another picture of all of us.
That's how
taken
we
about
look
a year
like.
ago. Yeah.
That's how we look like. We're not like stuck at home.
Yeah, exactly.
There
Yes, that's
are
everything. So yeah, thank you very much for listening. And yeah, we'll both be very happy to take any questions.
There are loads of questions trickling in already. So
Okay.
we have, for example, one that's come up several times is about the stem cells. Are these from an embryonic source, or what source do they come from?
So I can take that one. So as I explained, they are induced pluripotent stem cells. So that means that we take a skin graft, for example, from a patient, a few cells from the skin or any other tissue. And these cells, we grow them in special conditions so they can be grown back to the stem cell condition and pluripotency. By including in the media these different growth factors and components that they will trigger the cell to change, sort of like reprogramming. So they're not embryonic source, they are just sometimes from skin or like mouse swab. And once they are changed back to this stem cell state, then we can change them back and differentiate it into any type of cells that we need to study. In this case, we were showing neurons.
And how do you extract the tumor cells from patients?
Yeah, I'm happy to take, or yeah, both. So a lot of these tumor samples will be taken at the surgery stage from patients. So they won't be specifically for our project. They'll just be sort of an add-on to the treatment that patients will already be receiving. So yeah, surgeons will remove any tumors that the patient will have, and then they'll send some on to us, samples of that onto us for further use.
And how do
And
you
this is all with consent and everything, of course.
Yeah.
And how do you distinguish the cancer cells from the healthy ones?
I'll pass one
That's
to
very
Maria because that's a good one.
That's a very good question. Because of the conditions we grow, the cells are optimum for cancer cells. So healthy tissue will die eventually. All these healthy tissue cells will die eventually because the conditions we put in the media, they're not optimum for healthy cells. And that's the way we select only the cancer cells. Because at the beginning, when we derive a biopsy, and a biopsy can be, can contain healthy cells, can contain connective tissue, and obviously the tumor cells. So all this connective tissue, the fibroblasts, they will disappear eventually as well from the media because of the conditions of the media.
And how long is your average working day before COVID?
You want to answer that, Adam?
Yeah. Yeah, before COVID, yeah, I have to remember now. It's a typical 9 to 5 sort of working pattern, lunch breaks. But it does vary. It's true. So there might be some days where we're in the lab for 1 hour and then we can catch up on office work for the rest of the day, or other days where we're a bit busier and we'll be in the lab for a larger portion of the day. It does vary, but it's not working overnight or anything like that. Although Monica will probably disagree with that.
Monica will disagree with that.
Yeah. But yeah, generally speaking, it's a standard 9 to 5 job day like any other, really.
Yeah, I have to say, but because our job is not directed, related with the donors, because as we say, the biopsies we get, they are from cancer patients that they go undergo surgery. So the surgery is programmed and is planned and everything. So we know when we're gonna get a sample. But yeah, like Monica, she works in the HCA project, so they get donors any time of the day, any day of the year. So they have to be ready.
Sorry, Elena, we've got a few questions in the chat box here as well. One of my favourite ones is, why are your lab coats blue instead of white?
That's a Sanger thing, I think.
Yeah, I have no idea.
I think because the Sanger code is white lab codes, they're using CL1 labs, which they are like the lowest risk. So basically, when you work on the bench, you can work on the bench. You don't need these microbiological safety cabinets, and you don't work with any biohazards or anything. And the blue one is for CL2, and I think orange is for CL3.
Yeah, generally, yeah. We also have different lab coats if there's visitors coming, so you can know if there's someone just visiting and they might not know where they are. Or there's other lab coats if we're in a different lab where we're not supposed to cross labs without changing, just in case. But yeah, blue, yeah, I'm not really sure why. Yeah, it must be the Sanger, yeah, colours.
So someone else, so Jessica's asked, how do you work out which media is best for your cells? Is it through experimentation?
Yeah. Do you want to answer that? I can answer, I don't mind.
Yeah, I don't mind. It's up to you. You can go for it.
Okay. So most of the projects we run in SIGA, because we are a core facility, we work with already established protocols. And these protocols, they describe the media type for that type of cell. We do some R&D improvements with the protocols because, because of the larger scale of the science we deliver, we need very robust protocols that they're gonna work 99% of the time, for sure. So we get a protocol and then we develop a bit further. But usually we have an idea of the media and the components our cells need.
That's great. There are still loads of questions on, but we need to move on because time is running. So thank you very much for those great questions. We will now pass on the stage to Anna and Monica, who will take you through some parts of the Human Cell Atlas project.
Hi folks, so my name's Anna. So yeah, I work on the Human Cell Atlas project at Sanger. Behind me is a picture of the lab I work in. If I was actually there right now, I would be PPE'd up like Monica is, but it's slightly more messy perhaps than CGAP's lab. So I actually work primarily in a different building, which was the Morgan building that Elena mentioned, but we do a lot of work with the CGAP folks who run a lot of what we call the pipelines. They run a lot of processes for us. So if I perhaps just first introduce myself, as we've got a lot of students, I thought that might be interesting to you. So I'm a wet lab scientist, which means I work in the laboratory. So I work with tissues and cells and chemicals and doing experiments. A lot of my colleagues are dry lab scientists, which mean that they do computational programming and coding and analysis of data. So there's a whole kind of field in studying that, which I'm sure there are other people at Sanger who would be happy to spend a long time talking to you about that if you want to know about the dry lab part of the science. I was educated in the UK, so I did my GCSEs and A-levels here, and I went to university at Bath to do biochemistry. And if anyone's thinking of going into wet lab science, I did 2 6-month placements, and I think doing placements is invaluable if you want to go into science. It's a really useful thing to do. One of those was at Sanger, and another one was in America. So also if you want to travel, science is a great thing to do. After I'd finished my degree, I did a PhD in molecular biology, and then I worked in the pharmaceutical industry for about 7 years before I started here at the Sanger Institute. So I thought, as a way of introducing you to the HTA project, I'm first just going to play you a little video. So let's just see if I can share my screen and see if I can play this. Let's see if it will come up. Oh, is it going to play? Sorry, I've got you guys in it. There we go. It will come in a second. You should be able to see a picture of the world. There we go. It takes a while to catch up.
The human body is comprised of trillions of cells. Even though cells are the most fundamental units in our bodies, we know surprisingly little about them. For example, we still don't fully understand how many cell types there are, where they live, and which genes are active in each cell. This greatly limits our ability to understand biology and treat disease. But scientists now have the tools to find out which genes are at work in individual cells, and they can do this in thousands of cells at once. Still, with trillions of cells in the human body, it would take decades for any single team to do it all. This is why scientists from around the world have come together to build and share a comprehensive reference map of every cell type in the human body in a project called the Human Cell Atlas. When geneticists can identify disease-causing genes, this map will lead them to the cells where those genes are active. And when scientists develop precision therapies that attack disease cells, the Human Cell Atlas will help them strike the right targets. This atlas is the key to understanding human biology and the development of disease at every level, from molecules to cells to tissues. The Human Cell Atlas will impact almost every aspect of biology and lead to a richer understanding of the most fundamental units of life.
Okay, so to perhaps just elaborate a little bit more on that video in terms of what we are doing. So basically, as I said, Human Cell Atlas is looking to, well, the video described that the Human Cell Atlas is looking to basically try and catalogue the different cell types in your body. But it's more than just a catalogue. We want to build a map where we look at the spatial location of those cells as well. So I would hope that in a few years, because this project is a long project, it's going to take us 10 years or so, it's going to take a long time, that we'll get to a point where there will be a website with a human body, and you'll be able to click on a tissue in that body and zoom into the structures in that tissue, because it's very important that every tissue has a structure. It's not just a bag of cells, it has structure, and that structure is important for its function. Then you'll be able to go down further to look at what the individual cell types are in the different regions of that tissue. You'll be able to understand what genes they're expressing. You'll be able to get an idea of what those cell types do, and also how they interact and talk to each other, because cell types do interact and they do talk to each other to do their jobs. And then ultimately, understanding all that will help us to understand how those cell types might be changed if we compare against a disease, and how we might want to think about treating diseases. So I think that this work could be really quite fundamentally useful to understanding basic biology. And our hope is that in the long term, it will have an impact on you, because we hope that eventually it will impact on medicine, it will impact on diagnostics, so it will impact on you. So in terms of our cell types, really, as that video said, we know kind of less than you might think about cell types, and you might want to go have a look on Wikipedia for all the cell types in your body. And I think there is actually a page with how many cell types there are in our body, and I think it's got something like 300 or something. But from the work that we're doing, if you look at just take one structure, so take the retina, which is at the back of your eye, that is the surface that receives light and converts it into electrical signals in your brain so that you can see. That structure on its own has got over 100 different cell types. So it's not quite as simple as Wikipedia would say. And if we understand what our different cells are, then that helps us to understand the biology behind how we're built, and also how our organs are built from cells, and how that makes our organs work, and therefore how you are alive and how you work, and what goes wrong when you get a disease. So we hope this project will help us with understanding that basic biology. And then once we have a better understanding of that basic biology, it can help us to understand things like why some medicines may have side effects. So for example, if you developed a drug to target a tissue or a cell type that's in your kidney, and we have this big atlas and we say, oh, well, that molecule that you're targeting, that's also in the liver. That gives us an idea that maybe we might have side effects in the liver. So it's useful for that kind of thing as well. And it's also useful, for example, perhaps it could be useful in the future for developing diagnostic tests. So we have simple tests like blood tests now, which look at a limited range of cells in your blood. But with this atlas, once it's fully generated, we'll be able to look at all the many different cell types in your blood and know what a normal human kind of blood sample really looks like, and then be able to compare that against you. So I think there's a lot of potential uses to this project once it's kind of reaches maturity. So it's quite a long-term project. It's also quite a big project in that, although we're talking about the work that's being done at the Sanger, this project actually is a worldwide project, and we've got collaborators all across the whole world. The main heads of the project are Sarah Teichmann, who's at the Sanger Institute in the UK, and also Aviv Rajiv, who is at the Broad Institute in America. But as I say, we've got scientists all over the world who are working on this project. So it's quite a large project. In terms of what we're actually doing, there's really two types of data that we're generating on your cells. So the thing that kind of spurred this project to start was the development of a new technology that's called single cell genomics. And what that means is, previously, if you wanted to look at what genes were being expressed in a particular tissue, you'd take a lump of that tissue, you'd literally stick it in a blender, and you'd look at what genes were expressed in that piece of tissue. But that piece of tissue could contain hundreds of different cell types, and you just kind of get a homogenized result. So you don't really get the detail of the breakdown. Whereas now we have the ability to take that piece of tissue and do what Maria was showing, where you can break it into individual cells. And now we can look at every single cell, and we can look at what genes are expressed in that cell. And that allows us to distinguish in that piece of tissue which cells are different, what different cell types are there in there. So then we can start to look at what those cells might do and that kind of thing. So that uses the kind of cell culture techniques that Maria was showing in CGAP. And CGAP, at least pre-COVID, were doing work with us where we'd receive tissue samples and then we dissociate them into individual cells, and we do this analysis where we look at what genes each cell type is expressing. Then the other side of the work that we do is what we call our spatial pipelines, the spatial technologies. And that is understanding where those cells fit in 3 dimensions. So like I mentioned, each of your organs is not just a bag of cells where the cells float around. Rather, the tissues all have a structure that's very important for their function. And then we want to understand where the different cell types sit in that structure to help us to understand their function. So that's what Monica's going to show you a little bit of some of the equipment that we use for that in the lab, speak a little bit about that. So I guess I will pass over to Monica, if that's okay now, to show you a little bit in the lab.
Yep, thank you, Anna. Hi guys, I welcome you from actually in the lab. My name is Monica, and I love all the questions already. They're so in detail. Some of them are really, really amazing. So I studied in London. I studied in Queen Mary zoology with genetics. So I think if you would like to work in the lab, I think the university is the way to go, and especially one of the biology courses. And it was amazing fun. So I do recommend it to anyone. I did have a great time. So I work in CGAP, but specifically for the HCA project. So we basically here in the wet lab, we are helping the team with my old team to generate the data for the HCA project. And we work doing it a specific type of science, which is called histology. So you did see some of the movies already. I'll try to walk you through the lab, and especially my part of the lab. So I'm hoping I'll be able to share it with you. Please bear with me. If I turn my camera, can you see it? So if you walk in, this is our histology area. So you saw some of the equipments already. So this is a cryostat that you showed the movie. I work there almost every day, cutting and collecting tissues and cutting it into very small and very thin slices. The slices are usually between 10 and 12 micron thick, so they're really, really thin actually. And the temperature inside the cryostat is usually -20. So can you imagine sitting with your hands inside that machine for almost half a day with -20? It might not be really nice sometimes. The next machine that we use as well, it's called Leica Bond. So yes, we call it really cute. We call it Mr. Bond, and we work on it. It's absolutely amazing machine. It helps us to see the RNA inside the cells. And you showed this short movie about how Sophie was loading it. Initially that protocol was taking around 2 days. Thanks to this machine, it was cut short to only 6 hours that we run overnight, which is a big help in the lab. We also have on the other side some of the dissecting microscopes. So this is very specific microscope that I use to dissect the tissue, to cut it in a specific way to specific size, and you can see the tissue on the screen as well while you're cutting it. And it helps you to see everything with the detail and highlight it with the lamps. We also use different microscopes. So this microscopes is, for example, for checking the slides. When you cut the sample on the slide in the Cryostat, you can quickly check if you got the perfect morphology and where in a tissue you are. And we also have our scanner. So this is the fluorescent scanner. On the top, you can see the fluorescent lamp. And I will show you in a second. I'll try to share my screen now, if that's okay. So as I mentioned, can you see this? Bear with me. So as I mentioned, we use a part of science that is called histology, which basically helps you out to check morphology and the structure of the tissue. And to do it, we use different stainings, but at the moment, I'll just show you the very basic staining, which is using hematoxylin and eosin. In a short, we call it H&E. And we can produce some slides that look like that. I don't think you can see the presentation. Can you? No. Do we know why we cannot see the presentation?
No, I don't know.
Okay, I'll try to stop sharing and try it again.
Mm-hmm.
Sorry about that. So I'll try again. I'll do it without. Okay, so can you see it now? Perfect. Okay, so this is the image that we get when we do the H&E staining with different types of tissues, and most of them look absolutely beautiful. I don't know if you will be able to check to actually guess which one is which one without the titles on the bottom, but you have example here of the thymus tissue, of the intestine tissue, and one of the airways as well in the end. The airway is one of that tubes that is inside the lung. So this is the first part that we do for the H&E when we check the tissue morphology. In that machine that's called Bond, we do the fluorescent staining, and we do the protocols that they called RNA scope. So this is the different method that we use to see what kind of RNAs are inside the cells. So the machine helps us to stain RNA with specific dyes that they are called fluorophores. And then thanks to our fluorescent imager, we can take a picture of that cells, and we can see exactly which parts of the RNA, which genes are actually flashing and giving different colors thanks to that fluorophore dyes. So the images after that, they look something like that. So this is one of the example that we do sometimes run our positive controls, so the controls to check if the microscope works correctly. So, for example, you can see the thymus and all of its morphology, but also when you stain the same tissue in an RNA scope, this is the image that you get, which gives you the morphology first and then RNA expression second. And if you would like to compare them, you can see the same tissue with the two different protocols, two different methods, and that will actually help you out to see where and when in the tissue, so the spatially, when the RNA expressed and how. So if you have any questions to me, probably that's it for me at the moment. But I think we have questions later on, right? And I will pass everything to Anna, then I stop sharing.
Thanks, Monica. So I was just going to show one more RNA scope image just because it's like one of my favorite images, because it's one of the first ones that I generated when I was starting to set this method up in our lab. So let me just share that and I'll explain to you what it is. Can't make that full. Oh, I'm trying to make it full screen, but Zoom's in the way. Hang on. Well, you can see the image anyway. So no, next one. Here, this one. So you should be looking at an image which is basically blue, yellow, and red. And so this is a piece of uterus tissue. And when this project was run by two of my colleagues who work on Human Cell Atlas, Rosa Vento and Mirjana Efremova, and they're interested in studying pregnancy. And so this is a sample of uterus, as I said. And when they started this project, this particular region of the uterus was thought to be one layer of cells. And what they did was they took some samples of uterus, they broke it into individual cells, they looked at what genes were expressed in the different cells, and they realized there's more cell types here than we thought, and we'd like to understand how these are localized in space. And they hypothesized that they would be in layers. And so they asked me to do this staining. So the blue dots are nuclei. So every blue dot is a cell. And then the yellow dots are RNA, which is gene expression for one of the cell types they were interested in. And the red is a different cell type they were interested in. And you can see this proves quite nicely that indeed the cell types they were looking at are in layers within the uterus tissue. And these different layers express different hormones and have a particular function during, I think this was non-pregnant samples. So this was just during the development of the normal human uterus. So it was quite interesting and important work. And this was a new discovery by them. And I just quite like this image as well, because it's just a very pretty image to look at. So one of the nice things about the spatial work is it's just beautiful to look at. So I just wanted to share that. And that's just an example of some of the spatial work that we do for HCA. And then I just wanted to finish with one last thing, which is kind of a plug for a competition we're doing. So for the students who are out there, if there's anyone who's years 7, 8, or 9, or for any of the teachers, we're going to be running a competition in January, which starts on the 8th of January. We're going to launch it with a CPD session for the teachers. And the idea is we're asking year 7 to 9 students if they would try to design a card game that's based on the Human Cell Atlas. And it should be quite exciting because the winning game will actually be produced. So the team that wins will work with a game design company, and you'll actually get to work with them to finalize your game and then get packs printed, which will be sent to all the participating schools. So if anyone's interested in participating in the game challenge, for teachers, there's an email address here, which is Susie, who's running the competition. You can email her, she'll send you information. Or you can look at the website that's at the top. It's hosted by Little Inventors, and they're running this challenge for us. And then the entries have to be submitted by the 26th of February. So I just wanted to plug that competition for everybody. And that's all I have. So.
Thank you very much. That's great. We have loads of questions. I can't even keep track of all of them. So, for example, there is a question about cancer cells, whether cancer cells are within the scope of the HCA projects, or whether they are considered part of the human body.
So the HCA project is studying cancer. The majority of what we're doing is looking at normal tissue, because we have to build that baseline first. But there is a distinct section that is looking at cancer. And are they considered part of the normal body? Well, they're a disease state, aren't they? They're a state of cells that have had whatever happened, mutation or whatever's happened to them to cause the cancer. Does anyone else want to pitch in? Or is that okay?
Yeah, so in the Sanger Institute, there is a whole program called Cancer Mutations and Somatic Mutations and Aging. And they study precisely those processes that where changes in DNA cause degeneration of a normal cell into a cancerous cell. And they are trying to pinpoint and identify what factors in environment or lifestyle or whatever are causing these changes. Another question is, have there been any surprises discovered by single-cell genomics so far?
So there have been many. There have been many new cell types discovered, but I guess one, well, let's say a couple that come to mind. One is one of my colleagues, a gentleman called Felipe, and also some other colleagues in another part of the world identified new cell types in the lung, which are called ionocytes. And they're quite interesting because they express a gene that's involved in cystic fibrosis. So that's quite interesting. And they're following that up to see if that cell type is, contributing to cystic fibrosis, if that cell type is involved in the disease. So that's one thing. And on cancer, some of our other colleagues at Sangha, Sambajati, works on pediatric cancer, so that's cancer in children. And he's been doing single-cell genomics on tumor samples from children and also on healthy samples. One of the things that they have identified is that one of the tumors they look at, which is Wilms tumor, they think that they have identified that the origin of that tumor is basically aberrant kind of fetal development that kind of keeps going, and those cells should have stopped and they should have differentiated, but they don't. They've turned, and so they've produced a cancer in the young child. So I think that's quite an interesting finding as well. And there's many more, but that's a couple to name.
How have you split up the work on the Human Atlas project between the research labs around the world? This is collaborative research.
I think it's largely by tissues, because different labs get involved because they're interested in the lungs or the kidney or whatever they study. And then you also have labs like Sangha is a little bit more general. We're more focused on the technology. So we are very good at doing single-cell genomics. And it's basically then getting all those different people with different expertise together to work together and make this whole project work.
There's a couple of questions from the chat box as well. So someone says, how do you study the behavior of the cells in their natural environment?
So this is a challenge. We can't really in humans. I mean, obviously you can look at animal models, but they are not perfect. Or you can do what the CGAP people are doing, where they're looking at organoids. So you can take cells out of a person, turn them into organoids, and then try and study those cells in a dish.
And how has your work changed the way you think about the matter of your own body? I mean, anyone can pitch in and answer these questions. I'm kind of conscious of throwing all of this at Anna, but this can be a conversation. It doesn't have to be.
Definitely showed us that it's more complicated than we really think. And the more we work here, the less we actually realize, the less we know. So the more you work in the lab and discover stuff, you actually, the more questions are coming after that. And yeah, so it's not as easy as you would think.
And another interesting one is, given the variability in terms of human genomics, how do you decide what is normal?
Oh, that's a great question. We have a whole project at the moment just specifically dedicated to deciding what normal is, as Elena will mention. Yeah, so for the Human Cell Atlas, we're starting with what we can get access to. But ultimately, the idea will be to have a range of ethnic diversities, a range of ages. But obviously, that can be challenging because, I mean, for example, with donor samples that we take from organ donors, they are often from older people. You don't get so many from younger people. And you are somewhat limited by what you can get access to. But the goal is to try and introduce that diversity. And people are thinking very carefully about that, on how to introduce that diversity and make sure that we are covering that range of diversity.
Louise, do you have, can you?
There's one from Jessica, it's one for the CGAT team, I think. Why does one of the doors in your labs have lentivirus written on it?
I was just writing a reply to that. Yeah,
I was typing that one too.
Oh, okay. Yeah, so we use lentiviruses in the lab. The lentiviruses we use are completely manufactured. they'll be used to introduce genetic material into our cell lines. So, for example, if we've got a cancer line and we want it to express blue fluorescent protein to analyze it, we'd infect them with a lentivirus that contains the genes for BFP, the blue protein. So it'd integrate its DNA into the cancer lines, and then the cancer line would be genetically modified. So yeah, we use them as a tool. It's not like a yeah, they're very useful tools for introducing genetic material into cell lines.
Any other questions that can be, well, I have a question on the Q&A asking someone to play the guitar for us, and I suppose that's directed at Louise, who has the guitars behind her. But more to, there is also a question about Monica. You did mention what you studied and what you're specializing in, but if you could talk about that a bit more. There's a question about that.
Oh, okay, yeah. So I'm originally from Poland. So I arrived in UK and I decided that I would like to study. I was always interested in biology, especially zoology. So I applied to a couple of universities and Queen Mary accepted my application. So I studied there for 5 years and I did my master's there as well. So I'm not sure what else to say. I mean, the course was absolutely amazing. I do recommend it. I do recommend any courses in UK. Education in UK is really, really nice and interesting. So I enjoyed, like, for example, field trips. Oh, if you join and you can go for the field trips with the university, we went all around the world. We went to South Africa, we went to Burma to study different species of animals. So my specialization was with genetics, so I also did it on the more about the genetics as well, but with animals, which is maybe not as detailed and like in humans, but it still helped me out to get to laboratory and work in here. So
there are another couple of really good questions that are, does everyone need computational skills to do these studies, and are there specialists within the teams?
So I could just say to that, I'm afraid I struggle to use my phone. So no, there are specialists within the teams. About half of our team is dry lab people that analyze data and the other half is wet lab. If you can do both, that's great, but it's not essential.
And there was another question. There are so many, I can't keep up. I'm a bit, so, oh yes, do you think there should be an opt-out system for collecting stem cells from cord blood? Thinking about, you know, disease and stem cell therapy.
Well,
in the
it's
future.
a very wishful thought. Like, you know, if they all, you know, when we are born, just take a little bit of the cord and keep it, you know, for further. But the problem with that is the maintenance, is how expensive it is to maintain those tissues frozen and, you know, and ready to be used whenever you need it. So that's the main problem, is money, basically.
Yeah. Well, and also, and this is from me. So how certain can it be that you preserve these cells over long periods of time? Because it's not a given that all of them will be able to be recovered after storage. There's another question about if there is a cell atlas for simpler organisms than humans.
Yes, of course there are. There is the malaria, there's mouse. Those are the two main ones I think of, but there's, yes, people are making cell atlases for many, many organisms. Yeah.
And I can talk about the malaria one a little bit simply because that's kind of my subject. But but mainly because malaria is a single cell organism. So here we're not trying to identify different cell types, obviously, but we're trying to identify the gene expression of the different stages of the parasite that change quite dramatically throughout the life cycle. And that knowledge helps enormously to try and find drugs, targets for a vaccine and treatments in general. I think we've gone over time, and I am thrilled to see all the questions, and that's to see that people are still with us. So I'll give a few more minutes for a last round of questions before we end this fantastic session. So if you have any burning questions, fire away. This is your chance. Oh, there's a question saying, will there be another webinar like this? Well, yes, we hope so. And as there are no more questions coming in, I think with that, I will thank all the scientists who have come and given us their time. It has been live as much as we have managed to do it, because of course under the present circumstances is not always possible. And we thank you very, very much for joining us today. There is a little survey at the end. Please do fill it in, because it helps us modify and improve these seminars over time. So do please give us your thoughts. And with that, well, I'm just going to wish you a Merry Christmas, and hope that next year is a better one. So thank you very much.
Thank you for having me.
Thank you.
Bye.
Bye.
Bye, everyone.